The next few slides that I have here, this I have explained, I think, in very great detail in terms of the the diagrams I drew, but we'll just go a little bit quicker here so that you guys are familiar with the content. So we talked about the inside the membrane versus outside, extracellular fluid versus inside the cytosol. About the resting state is negative 70 millivolts, and when depolarization takes place, it will go from negative to positive. How does it do it? It does it via specific channels, right? For the touch specific neurons, right? We said there is mechanoreceptors. So mechanoreceptors is like I could press on my finger here, right? To cause mechanoreceptors secrete. So we have sodium leaky channels, and we have voltage-gated sodium channels. We have leaky potassium channels, and we have voltage-gated potassium channel. What's the distinction? The distinction is this: the voltage-gated channels, regardless of what they are, they will only trigger when you hit channel-specific threshold potential. For sodium, I use, let's say, negative 50 millivolts. And when you trigger the threshold potential, only then the voltage-gated channels open. And they do so because they have these gates, right, that you have to basically trigger in order for the channel to activate. But on a leaky channel, right, if the channel is leaky, it's permeable. So you could have changes of these cations via diffusion. Diffusion is usually based on the concentration gradient. You want to go from high concentration to solution. And I have said multiple times, right, cells are bags of potassium and a sea of sodium. So inside the cells, always going to be high potassium. Outside the cells, always going to be high sodium. There's something else that I wanted to say, right? So we have a potential and we have graded potential. What is a graded potential? So imagine, let me just redraw quickly here. So let's say we have threshold potential is here. Have resting membrane potential is here. So let's say you touch, and I said this was sub-threshold, right? It didn't reach the threshold potential. Then you give me another touch, right? Maybe it's a little higher, right? But right before, right, you initiate it again, meaning before it goes back to the resting state, right? Let's say you do it in succession enough, right? You do it fast enough that what happens is they kind of build on one another. You want like this. And if enough sodium comes in to get to the threshold, we will have that action potential coming, right? So graded can be additive. They can come together to basically trigger the threshold potential, sodium channels open, and the action potential will essentially, right, sweep across the membrane, turning that negative to positive. It will do so, right, traveling axons all the way to the terminal side of it, right, postsynaptic cell, where that activity will lead to the release of specific neurotransmitters. So depending on what we're going to talk about, could be acetylcholine, could be norepinephrine, right? And then the information processing will occur downstream, right? So this is what happens. This is essentially all these diagrams. Was it Ajaya who was saying he finally understood how it all works, right? But they will show you, right, all the stuff we, plethora of time talking about this potassium exchange, leaving, coming in, depolarizing the cellular membrane, right, for all these to occur. I agree. I think they make these diagrams, it is a little more involved, but I think if you take it step by step, I think the material is explanatory, right? The other I want to talk about, we talked about voltage gated channels, right? We talked about ligand gated channels, right? So what is ligand gated channels? Ligand gated means you need to have a specific receptor. Here they have acetylcholine. So acetylcholine binds on the receptor side, and then only then this channel opens up. So once acetylcholine binds to the specific receptor, ligand-gated receptor, right? For acetylcholine, anyone remember the name of the receptor? What was it called? Helmi, your microphone is muted.
Nicotinic receptor.
Yes. Right. So here, the voltage-gated channels, what's so specific about them, and you want to keep in mind, right, for chapter, for cardiac chapter, is that the voltage-gated ones, they have two gates, right? So I was drawing it like this, right? I was drawing it like that. And there's an activation gate here, and there is an inactivation gate here. It's two gates. If one gate remains closed, even though the other one is open, the channel is inactive, meaning for it to have exchange of ions, the channel must be open. So if, let's say, the channel is in this state, right, can come in because activation gate remains closed. If, on the other hand, the channel is in this configuration, activation gate is open, but an activation gate is closed, again, no sodium can entrain. Why? Because an activation gate is closed. So this becomes, right, in the electric gradient changes, right, what opens and what closes. So for the sodium, what triggers the opening of this is reaching threshold potential, right? What causes the closure is once, right, it almost goes to its equilibrium potential, is what causes the closure, right? Almost reaching the equilibrium potential. When the channel remains open, you have an exchange of sodium coming in. So here we have the opening of both gates, right? Activation and inactivation gate. Here we see an activation gate is, so even though you have the sodium outside, they cannot come in of this purpose. And that's specific for the voltage-gated channels. Here, this one, I was referring to the mechanically gated channels. They're leaky channels. So this was the receptor, right? I said I touch my finger and the sodium comes in. So we see channel is closed, sodium is outside, but the moment applied pressure is done, right? So let's say stimulus came in, and the stimulus was not sub-threshold. So, right, the stimulus that comes in order to activate these channels must be strong enough, right? If this was the threshold potential, the stimulus cannot just terminate here. It either has to be, summative enough to reach threshold, or the applied initial pressure has to be strong enough to cause this. If it doesn't, it will not trigger opening of these channels, right? So here we see pressure removed. Yes.
Is this why we have so much stimuli on our skin all the time? This is why we don't react to it all at once, right? Like why we don't feel our clothes on our body.
Exactly. If any of you have a watch on their hand, like a wristwatch, right? Right before I said this, no one felt the pressure of the wristwatch. The moment I say any of you have a wristwatch, all of a sudden you feel the wristwatch in your hand, correct? Right? So it's, yes, a lot of these things, your body will essentially not actively send signals to, even though they're there, right? And you may not feel it, is because subconsciously, right, there's other stimuli that take precedence. But yes, the moment I tell you about it, right, all of a sudden you feel it. You feel, your wristwatch on your hand. But those are also, right, mechanoreceptors, and they're operated based on the amount of the applied pressure. And as we said, right, the strongest does not mean, right, you have higher amplitude. What it means is your frequency will basically increase until the impulse is removed, right? Right, and it's no longer there, right? So that's this here is mechanically gated channels. Usually they're leaky channels. The next thing we talked about, right? This was distribution of channels in the neuron. This was more particular when we spoke about calcium mediated exocytosis. Remember I said for the muscles, right? Calcium voltage gated channels came here. What action did calcium do specifically for the presynaptic cell? What does calcium do on presynaptic cell? For the muscle. Anyone recall? So the voltage gated calcium channels on the presynaptic muscle, they call the vesicles that are filled with acetylcholine to come and bond on the surface. So it was calcium mediated exocytosis of acetylcholine, right? So calcium on the presynaptic cells was responsible for release of acetylcholine. Anyone remember what was the role of calcium in actin and myosin when it was on the actual mechanoreceptors? What did calcium do?
Can you repeat that question?
Yeah, so I said the role of calcium on the presynaptic terminal was acetylcholine, right? That's what we learned in muscles. But what calcium downstream, actin and myosin? Anyone recall what did calcium do there on the actual muscle filament?
Hey, Nick, you've been breaking up a lot. Can you repeat that one more time?
Okay. What was the role of calcium on the muscle membrane itself?
Does it open up the receptor?
So on the muscle itself, I remember it was binding to a specific location.
It binds to troponin.
Troponin, excellent, right? So yes, so on the muscle side, it was bounding directly to the troponin, and when it bound to the troponin, it moved the tropomyosin out of the way for myosin head to dock with actin. So what you want to remember, actions of calcium, right? On the presynaptic cell, calcium triggers release of the neurotransmitter. For the muscle cells, it's being acetylcholine. On the muscle site itself, right? Calcium binds to the troponin to move the tropomyosin out of the way. So remember the distinction in terms of calcium here, right? Here they show you essentially the axon hillock, the top portion, leads to the axon, and it depolarizes. As I was saying, it goes from patch to another. So let's say if this was one patch, this was another patch, right? And it moves from patch to patch, using depolarization and repolarization to occur, right? Depolarization, repolarization. Once it does that, it goes to the resting potential, right? Once it reaches resting potential, right, it can either proceed to the next stimuli, right, or it can hyperpolarize. Hyperpolarize means that potassium is still leaving, right? It's leaving in excess until it returns back to its resting potential. Usually potassium wants to reach its equilibrium potential, but it closes before it reaches that point, right? And what you want to remember, depolarization is mainly sodium, mainly potassium. That is the major cations involved there, right? This is the propagation, the continuous conduction, right? How it travels from patch to patch, right? And sodium potassium pump, right? And what causes the equilibrium, right? So as it's from patch to patch like this, remember I said on your cells, on your neurons, we have sodium potassium ATPases. They're powered by ATP, which is mitochondria. Their goal is to get rid of 3 for every 3 potassium they bring in. So this is actively working in your cells. About 20 to 25% of your ATP is used in potassium ATPases. And the whole job is to restore that gradient, this electrical gradient that we see, right? As I said, right? And some cells are, right, bags of potassium and a sea of sodium. So these sodium potassium ATPases that are found in the membranes of the cells, they're actively working to throw away 3 sodium and bring in 3 potassium, right? And we know that to make ATP, you need oxygen, right? You need oxidative phosphorylation. So in a setting of shock, right, when you have low oxygen supply and your muscles and your cells in the body lack, right, oxygen, do you think your sodium potassium ATP are functioning well?
Oh, no, it's low oxygen.
Yeah, lack of oxygen. So not only you got to think in terms of there's hypoxia to the tissues. You also got to think of the processes that we have in the cells, right? What's bringing potassium in, what's bringing sodium out. So if a lot of these sodium potassium ATPases are stopped functioning, right? Perhaps you start to develop hyperkalemia. They are not working. They're not bringing this potassium. You have a potassium outside the cell now happening because of inaction of these sodium potassium ATPases due to lack of ATP formation, right? Because you're in a state of shock and you have less oxygen coming in. So a lot of normal physiology get distorted, right? And these sodium potassium ATPases are critically important for you guys to restore that balance between these cations. Right? We talked about this, I think, in depth, right? That I think this should be very clear, right? I'm going to go to this run here. This is, again, I'm going to explain it in a relatively faster pace because I think at this point, I think it should be very clear cut, right? So this was the synaptic cleft. Here we have acetylcholine release, right? And the exertion of the sodium influx, which then propagates down, right, to the motor end plate, causing, right, its effects by depolarizing this membrane here. Now, something that I want to explain, and you guys need to be very much familiar with, right, is these enzymes. So acetylcholine esterase. We said acetylcholine esterase job was to basically chop up acetylcholine, right? So acetylcholine esterase enzyme chops up acetylcholine, these receptors, it turns it into choline and acetate. And choline can get reabsorbed, right, back into the presynaptic neuron to make more acetylcholine neurotransmitter. The function of acetylcholine esterase is needed. Why? Because you have contraction of the muscle, for the muscle to come in to contract the muscle again, right? This enzyme must break up this acetylcholine so the next one can be released and work. We also had a condition, myasthenia gravis, right, where you have autoimmune processes that cause release of antibodies that basically attach on these receptors, and they don't allow acetylcholine to bind, right? So you remember what class of drugs we would give to those patients who have myasthenia gravis?
Physostigmine and hyoscyamine.
Yeah, that's the name of the drug, neostigmine or physostigmine. But what class of drug is it? How would I categorize the drug?
Acetylcholine inhibitor.
Inhibitor, excellent, right? So help me say choline esterase inhibitor. So if I give you a drug that inhibits this enzyme, right, you have more acetylcholine now that's found in the synaptic cleft, so it can exert its effects. So if I block this enzyme, less acetylcholine gets chopped up, more of it to exert its effects, right? Very good, right? This is presynaptic neuron, and this is the postsynaptic neuron. So I want you to understand, right, in the nervous system, we have presynaptic neuron, we have postsynaptic neuron, and in some instances, you will see that the postsynaptic is the one that's going to exert its effects on the target cell. And we will see that there's 2 neurotransmitters that are going to be involved. So I want you to be very, very clear in terms of the role of acetylcholine, right, for the muscle portion of it. And we're going to learn about very shortly about 2 neurotransmitters that deal with the autonomic nervous system, especially the sympathetic nervous system. right? Right. So it was the summation aspect, right? If you have a first stimulus, it doesn't threshold potential, but the stimulus is strong enough to reach the threshold potential. The moment that is attained, then you have the propagation of that actual potential going forward. Right. So now we're getting more and more towards talking about these mechanisms. So here you have the presynaptic neuron and you have the postsynaptic neuron. We're going to learn, right, in the autonomic nervous system, especially for the sympathetic nervous system, synaptic neuron here, this portion is going to secrete acetylcholine, but the postsynaptic neuron is going to secrete epinephrine. So usually, if you want to remember, the presynaptic neurotransmitter for the most part is going to be acetylcholine, for the most part. For the sympathetic system, the postsynaptic neuron will mostly secrete norepinephrine. And the norepinephrine is then going to exert its effects on the target tissue. We're going to learn what those are, right? So here we have a lot of these neurotransmitters that are in your body. They're released normally under specific stimuli. Now, if you understand how these work for your normal response, then you will understand how the response of the medications will work, right? So we talked about acetylcholine, right? And how it's effective for the muscle contraction specifically, right? For the system, it's responsible for the neuromuscular junctions, right? Autonomic ganglia. The norepinephrine, we're going to learn, it works through G protein coupled receptors. So the mechanism of actual response of it is a little bit different than we learned in terms of acetylcholine. Where this neurotransmitter found predominantly, this is found predominantly in your sympathetic of the peripheral nervous system, right? And when it docks to the specific receptors, and I'll show you what those are, we see, for example, heart, we see increase in heart rate. When it comes on bronchioles, we have bronchodilation, right? When it comes to the pupils, we have dilation of the pupils, right? So norepinephrine, another name is adrenaline, it exerts the following effects. Sometimes they will be called catecholamines. Why they're called catecholamines is because they have these catechol here, and it's derivative of tyrosine. So tyrosine being an amino acid. So they called catecholamines, right? Amino acid and the catechol ring. Epinephrine is secreted by the adrenal medulla. Remember I talked about there's that synapses to the gland cells. So neuron that synapses to the here being adrenal medulla is going to secrete epinephrine. Neurotransmitters work much faster, right? We talked about the communication between what? We have neurons that are myelinated. Myelinated neurons conduct this through saltatory conduction very fast. But epinephrine, right? When it's released, it's going to be carried through the blood. So it will be a slower conduct, right? Effects will be similar, right? When it gets to its target organs. And we're going to talk about what those are. The other thing that I want to focus on here, which will become important, is glutamate and GABA. Glutamate is excitatory, but the GABA is the inhibitory. I was starting to think of the medications, right? So let's go with the inhibitory first. So GABA. GABA stands for gamma aminobutyric acid. We see it here. This is one of the predominant inhibitory neurotransmitters. How does it do it? It does it by opening chloride channels, right? It's causing, right, the effects with negative chloride channels that open up, causing the membrane to become negative. We see these channels, right, specifically for your medications that you're going to learn, such as benzodiazepines, right? We have lorazepam, diazepam, Versed, midazolam. You give those patients during sedation, right? So when I give this medication and I want to cause sedation, sedation such as anxiolysis, less anxiety, hypnosis, I go to sleep, right? It does so by activating this inhibitory transmitter, right? Glutamate, on the other hand, is an excitatory neurotransmitter found in the brain. So glutamate is more on the excitatory end, stimulates it, right, via G protein coupled receptors. So why I have all this here, right? Once you understand the effect of the normal neurotransmitter or hormone response and what it does on the target cells, then you could kind of understand the effect of medications. So if I say this medication works by binding on GABA receptors, this medication is serotonin inhibitor, so more serotonin is available. This medication is sympathomimetic; it mimics the sympathetic system, right? Or this medication is a parasympatholitic; it blocks the parasympathetic system. So you could kind of understand the effects of this, right? We also have some nerves. We're going to talk about the ones I want to point your attention to, right? Specifically, that innervate the respiratory system, specifically C3, C4, C5 for the diaphragm, right? Why do I have this here? So in those demyelinating diseases, right, like such as we talked about, again, we talked about multiple sclerosis. If they demyelinate these nerves that myelinate towards the diaphragm, you may have a person having breathing issues, so we may need to place them on a ventilator. So we have different nerves that innervate different, I would say, locations. And in a clinical setting, right, you may see the doctor testing for them, right, with different responses. Right. So here we have the nerves in the arm, radial nerve, medial nerve, ulnar nerve. That's why in your trauma assessment, when you guys check for circulation, motor, and sensory, right, all three are distinct. How are they distinct? Circulation, you check the pulse, right? So I check my radial pulse. So I know there is perfusion to the distal extremity. See, right, I will elicit a painful response. Or if the patient is alert, I will ask, which finger am I holding? Why am I doing this? So when I apply my stimulus, I apply the stimulus to the mechanoreceptors. I just explained, right, how this propagates the impulse to the central nervous system, and it will interpret it, and the person may say, oh, I feel it, and you're touching my index finger. So his ascending pathway, right, ascending pathway, the afferent pathway is functional. Then you will say to him, you want him to do a motor response. You will say, okay, squeeze my fingers, or wiggle your toes, move your hands, or whatever you wanted that person to do. Okay, he squeezes your fingers. You know, the motor response is intact. How is the motor response intact? Right? Somatic system. You told them, squeeze my hand, right? So he's squeezing his hand after you asking them. So he's doing it under voluntary control, under the somatic system, right? Acetylcholine is released, synapses to the nicotinic receptors. The impulse propagate right down the T-tubules. Sarcoplasmic releases calcium. Calcium binds to troponin. You have now tropomyosin moving out of the way. Myosin head docking to the actin. You have muscle contraction. So now you kind of can picture, right? Why are we doing CMS? Why are we doing circulation motor sensor? We're doing this to give, like, you're not assessing for the fine motor movements. You're not a neurologist, but you're seeing that the big pathways, right? The big pathways that process the information to the brain, and from the brain to the effector motor neurons are working, right? And that's why we are testing that.